Hello friends, I am here with my channel Solving Questions with Gauri ma'am. Here we solve questions from various MSc entrance examinations. Let's have a question for today's video. The question is from JNU Biotechnology Entrance 2018. The question is, which of the following is mismatched? Oscillatoria, Cyanobacteria, TMV virus, whether it is a RNA virus, then E. coli is a photosynthetic bacterium, and the last one is a plasmid as a extra chromosomal DNA. So we have to find the mismatch out of these. So let's have a look at each and every option to find the mismatch. The first option is a Oscillatoria and Cyanobacteria. So this is a correct match. Oscillatoria is one of the example of Cyanobacteria. Other examples include nostoc, anavina, spirulina. These are some examples of the Cyanobacteria. So what are these Cyanobacteria? We also call these as a blue-green algae. These are a photosynthetic bacteria, and we consider these to arrive first during the evolution of the earth. These perform oxygenic photosynthesis, due to which oxygen has arrived on the earth and that is got useful for other life forms. So this is the photosynthetic bacteria. Then this Oscillatoria is a genus of filamentous cyanobacteria. It shows the oscillation in its movement. That's why the name is Oscillatoria. These reproduce by fragmentation, and we find these bacteria in a watery bodies, like in lake, pond. In water bodies, these can survive. The second option is TMV RNA virus. This is a tobacco mosaic virus, and the match for it, that is RNA virus, this is a correct match. It's an infectious agent to the tobacco plant. This is the virus which was first discovered, right? This infection of this virus causes mosaic pattern on the leaves. That is, discoloration of the leaves are observed after the infection of this TMV virus. So that's why it is called tobacco mosaic virus. And it is having a positive sensed single-stranded RNA in its genome. The next match given is a E. coli and photosynthetic bacteria. So all of us must be knowing about the E. coli. This is a bacterium we commonly use in our laboratories. The routine experiments for genetic engineering, for transformation, then for the isolation purposes in a microbiology experiments, we commonly use this bacterium, Escherichia coli. This is a gram-negative bacteria. This shows a pink color when we stain it with gram staining. This is facultative anaerobic bacteria, and this is found in a lower part of the intestine of the warm-blooded animal. When the lower part of the intestine, we find the E. coli in there. So these are not a photosynthetic bacteria, right? So this is the actually correct answer for our question. In the question, mismatch is asked. So this is a mismatch. E. coli is not a photosynthetic bacteria. The last option is saying plasmid as an extra chromosomal DNA. So this is again a correct match. When we say plasmid, it's apart from the chromosome. It's a DNA. This is a chromosomal DNA, bacterial chromosomal DNA. And in the bacterium, if the plasmid is present, it is present outside the chromosomal DNA. So that is an extra chromosomal DNA. This is a self-replicating unit. It has its own origin of replication. But for the replication purpose, it relies on the bacterial cell, right? In nature, plasmids can be transformed from one cell to another bacterial cell through the process of conjugation. Whereas we can transform plasmid using a transformation in our lab experiments. Secondly, this plasmid contains the gene which are used for some drug resistance. It may be for particular antibiotic resistance. Like it confers the tetracycline resistance, ampicillin resistance. If that particular bacteria is having the plasmid, then the bacteria becomes resistant against that particular antibiotic. So this is the property which is helpful for bacteria. Okay. Then here I have written some examples. PUC 18, PBr 322. These are the examples of bacterial plasmid. So the answer for this question is option C. That is E. coli is a photosynthetic bacteria. Because E. coli is not a photosynthetic bacteria. All other options are matching with each other. Time to have a question for my next video. The question is from TIFR-GS Biology Entrance Examination 2017. The question is about the enzyme. 3 enzymes are given and it is asked that what is the correct order of action of these enzymes during DNA replication. So helicase, primates and polymerase. These are given in 4 different sequences and which is the correct order in which these enzymes are. This is asked in the question. We will solve this question in my next video. Till then, don't forget to like, share and subscribe my channel and also to hit the bell icon. Thanks for watching.